You will remember yesterday that we talked about the first step in protein synthesis, and we said that was called transcription. And if you think about the basis of the word transcription, trans is a root that means across, and script means to write. And so really what's happening is that the mRNA is making a copy of the DNA that's in the nucleus. So I still have this piece of that up here on the board. Here was the DNA. We're inside the nucleus over here. And here is the mRNA, the messenger RNA, that after this DNA was zipped apart, this came in, these nucleotide bases assembled, and then they attached together to make the copy of the information that's needed for the protein. And now this mRNA is going to transfer outside of the nucleus. It's going to go out into the cytoplasm, and it's going to make its way to the ribosome. Because remember we said ribosomes are the places where proteins are synthesized. So let's take our mRNA, and we're going to take him out here, and we're going to move him to the ribosome. And I'm actually going to kind of turn it sideways here so that we can see it a little bit better. Okay, so this is in the ribosome. Now, while this has been in the ribosome, there's some more pieces of RNA that are already circulating out in the cytoplasm. And as you should have noticed in your reading, it takes 3 nucleotide bases to code for a particular amino acid. And so I've got a section of 3 and a section of 3, and in the DNA, those are called triplets. But once I get to the messenger RNA, each sequence of 3 is called a codon. Well, the transfer RNA now is going to match up the nucleotide bases, and for every set of 3, for this codon, there's going to be called an anticodon that then is going to hook up to a specific amino acid that's present in the cytoplasm, and it's going to bring that amino acid to the ribosome, and then the ribosome is going to hook everything together to make a protein. So let me show you again how that works. So on this piece of messenger RNA, the first section that I'm coding for is an adenine, uracil, cytosine. So what do I need to match up with those pieces? Well, remember, an adenine matches with a uracil matches with an adenine, and a cytosine is going to match with a guanine. So there are lots of different transfer RNA anticodons that have managed to pick up different amino acids, and that's what these pieces are supposed to represent, as amino acids. And so I have to find one that matches this code. And so this one here is going to match with this particular code. So it's going to come, and it's going to look like this. The next codon that's here is cytosine, guanine, adenine. And so what's going to match up with that? Well, I'm going to need a guanine, a cytosine, and then a uracil. And so that's going to be this one, and it's going to come into the ribosome, and it's going to come down here like this. Now, what happens from here? Well, remember, the whole point of this process is to assemble amino acids to make proteins. And so now these amino acids are right next to each other, and so you're going to have, again, another enzyme that's going to come along, and it's going to bond these amino acids together in what's called a peptide bond. Again, in our case, we're using Scotch tape. So we're going to bond those 2 amino acids together. And once the amino acids are bonded together, it's going to start to move away from the messenger RNA. And so that these can be recycled, we're going to actually break the bonds between the amino acids and the anticodons, so that these can actually go out and be used again. So when we break bonds here, I'm just going to use the scissors. So here's one tRNA that has been released to go back out and find another amino acid. And then here's another one. We're going to cut this one loose. Just like that. And so this tRNA can now go back out to the cytoplasm, and he can hook up with another amino acid and keep using them over and over. And now my amino acids have been hooked together into a protein. Now, proteins are going to be much longer than just 2 amino acids, but my board's only so big, and but I think you can understand the process as to what's going on here. Now, in actuality, the first codon that is used is called a Go sequence, and it does code for an amino acid, but there has to be a place where there's a stopping point as well. And so there are 3 specific stop sequences, which are also series of 3, and they do not code for an amino acid, but they just tell the tRNA, we're stopping here. This is the end of the line. We don't need any more amino acids. The protein is finished, okay? So again, this is simplified. It's much more complicated than I've shown you. The other thing I want to do here is I want to go through this other chart that you guys had. In fact, you had 2 charts. I'm going to go through those and make sure that you have all of that filled in correctly. So the difference between transcription and translation. Transcription is, again, where we're writing across. We're making a copy of something. Translation, you're probably familiar with that word, especially if you live in another country and you don't always understand the language, and so somebody sometimes has to translate, or maybe you use Google and Google translates for you to try and understand what that other language is saying. Well, out here we're actually translating a codon, an anticodon into an amino acid, so it's almost like putting it into another language. So transcription first, so we can carry it out of the nucleus, then translation to put it into the language of amino acids. So where does each one of these take place? Transcription takes place in the nucleus. Translation takes place in the ribosome. Okay, the nucleic acids that are involved, transcription is going to involve the DNA and messenger RNA. Whereas translation involves messenger RNA, because remember, it carried the code from the nucleus out to the ribosome, and then transfer RNA, which transfers the amino acids in the right sequence and then hooks them together. Okay? Now, what's the result of transcription? Well, the result of that is actually just a negative image of the DNA. Translation results in a protein. Now, one thing that I do want you to notice here, if I look at the transfer RNA that was actually used here, it was this piece, and then it was this piece. Those are the 2 pieces that we used. If I would go back and compare that sequence to what was in the DNA originally, you'll see that it matches up exactly. You can see all the colors match up exactly. There's a uracil in place of a thymine, but other than that, it's the same exact sequence that was in the nucleus for the DNA. So the reason for that is because we've got to get that exact code out to code for the protein, so therefore we have to make sure that we can make an exact copy. So that's why we need a negative image to begin with, because it's got to be transferred really twice. Okay? So what's the difference between DNA, mRNA, and tRNA? Again, this is a chart that you should already have filled out, so let's go through and check those answers real quick. So where does it work? DNA works in the nucleus, and it contains a triplet of nucleotide bases that are going to code for amino acids. It doesn't contain the actual amino acid. It is involved in transcription, but because it cannot leave the nucleus, it is not involved in translation, which happens in the ribosome. Messenger RNA is the one that's going to take the message from the DNA out to the ribosome where the protein is made. So messenger RNA works both in the nucleus and the ribosome. It contains a codon, that's the set of 3. It does not contain the actual amino acid, but it is involved in transcription and translation both. Both of those pieces are needed. And then finally, my tRNA, or my transfer RNA. It works only in the ribosome. It contains an anticodon. It does interact directly with the amino acids. It is not involved in transcription, but it is involved in translation. So you want to make sure that you have your chart filled in correctly. Now, you also had a coloring sheet that I gave you so that you could hopefully see yet another version of this. I think the more times you see it and the more different ways you see it, the easier it's going to be for you to understand. So you can see, if you get this out and put it in front of you while I'm talking about it here, you can see the ribosome, you can see the nucleotide bases all set up there. They had way more than I had. And so they've got 2 amino acids already there and attached. What I want you to do, can you figure out which of those amino acids is going to come next? If you look at the codon in the mRNA, you can see that that it has an A, a C, and a U. So what has to pair up with those? Well, with the A, I have to pair a U. With the C, I have to pair a G. And with the U, I have to pair an A. So U, G, A. Do you see any amino acids out there that are hooked to an anticodon that says UGA? Well, if you look, it's the one that's marked with a letter H. And then if you look down in the key to see what that H stands for, it stands for tryptophan, and that's the amino acid that's going to be next in the sequence. Okay, what about the last one? Can you figure that one out? You'll see that the last 3 codons in the mRNA are an A, a G, and a U. So what has to code, what are the anticodons that have to go with that? Well, an A goes with a U, a G goes with a C, and a U goes with an A. So I need a UCA. Do you see anything up there that has a UCA on it? That should be the amino acid that has a J marked on it. And then again, if you look down in the key, it says that J, that amino acid's name is valine. Okay, so those amino acids, or those coloring sheets rather, should hopefully help you better understand the concept of translation along with the one that I gave you yesterday. All right, so hopefully now you have a big picture of how a cell is going to use its DNA to code for proteins and how that message is going to get from the nucleus out into the ribosome where production actually takes place.